Hello. So here's an outline of sort of 3 topics that I'm going to talk about today. First of all, AgBiome is a lot smaller and a lot newer than most of the companies in the area. So I'm going to give an introduction to our company and a little bit about our business model of partnering with other companies. I'm also going to talk a lot about how we've built our microbe collection and then how we're going to exploit that microbe collection to discover new products for agriculture. And then the last thing, we've sequenced over 15,000 microbes from our collection. And so I'm going to talk about some of the early lessons that we've learned from sequencing those microbes. So here's just a quick overview of our company. We got venture funding in 2012, late 2012, from some standard investment firms, as well as some strategic partners, including Monsanto and Syngenta and Novozymes. We actually started operations in early 2013. We've grown to over 30, or right around 30 full-time employees at this point. And the product targets that we're looking to develop, we want to develop trait and biological products for fighting pests that attack agricultural crops. So as far as how we fit into the larger ag community, we are the discovery engine for new product discovery, and we'll be partnering with other companies to bring those products to market. We have multiple partnerships in place, including one that we recently announced with Syngenta. That's a trait research project, and we have other agreements that are in various stages of negotiation. So a little bit more about our partnership model. For certain products in certain markets, we can start with the screening, go through proof of concept, and on into the commercialization on our own. And that's a model that we'll be taking for certain biological products in smaller markets. For larger markets, we'll need to partner with other companies in the later phases of product development and commercialization to bring those to market. And then for trait product development, we'll be partnering early on with other partners to take those products to market, so at the proof of concept stage. And we have an LLC structure that fits well with this partnership model, so we have the ability to license individual projects into subsidiary companies, and we can sell those outright without having another company need to buy our entire company to have access to those products. And just a little bit about our mission. We had a branding exercise, and we decided that this was the mission. We have the ambition of one day being the biotech company that has discovered the most products on the market and also having the happiest employees. So we've heard a lot about the plant microbiome today, and one thing that we haven't talked about a ton is that the microbiome has a long history of generating products used in agriculture. So since the 1920s, the spore-forming bacterial species Bacillus thuringiensis has been applied as a whole organism to help fight pests in plants. And then starting in the 1990s, once it was discovered that cryogenes were responsible for that pest-fighting activity, those were engineered into plants and introduced into the market as transgene products. And as you've probably gotten a feel for today and from other talks you've probably heard, we're just starting to scratch the surface in terms of the plant health benefits that are possible to harvest from the microbiome. So some of the, we're looking to tackle some of the biggest problems in agriculture. So we have a program in disease control where we're looking to fight fungal diseases with things that come out of the microbiome. We're also looking into insect control, and we have programs in insect and nematode control. I wouldn't say we have a dedicated program for yield enhancement, but some of the information that we're collecting could serve as a strong foundation to start that program more dedicatedly in the future. So for us, our quest to harvest the microbiome for new products starts with our sampling. We go all around the Midwest to collect different samples. And so far, we've collected about over 2,000 samples, and so an example of an environmental sample would include a corn root or an insect of some sort. And we have agreements in place with seed companies and landowners that allows us to have unrestricted ownership of our microbes, and that's important for our partnering with other companies. It's important that we have unrestricted rights to the microbes. As far as where we're collecting these samples from, it's driven by our product goals. We want to make products for corn and soy, and so a majority of the samples we collect come from corn and soy, which are the blue and orange, and then we also collect from other sources as well. So once we have an environmental sample, we fractionate that sample into what we're calling individual collections. And so, for example, from corn roots, we can fractionate those into the endophytes and the rhizosphere. And the reason why we do this is sort of well documented at this point. There have been several studies showing that if you look at the phyla, the distribution of phyla that are present in, say, bulk soil versus the rhizosphere versus the endophytic compartment, that there are different constituents there. And certain species are overrepresented in the endophytic compartment while others are underrepresented. So that's the reason why we fractionate our samples, is so that we can increase the representation of endophytes in our collection. Otherwise, they'd be drowned out by the things that are in the rhizosphere. So once we have an individual collection, and I should mention we have over 8,000 of those so far, we can plate the collections out on media and then pick individual colonies to go on to isolation. And then we archive those microbes, those isolated microbes, as glycerol stocks, so we can go back to them later. So how are we using these microbes to discover new products? Basically, we take a microbe, we grow it up under different conditions, and then we screen it against one of our targets in either disease, nematology, or entomology. And then once we have an active microbe, we ask the basic question, is the activity conferred by a single gene? If the answer is yes, then that gene is a good candidate to enter our trait product development. If the answer is no, if it's something not encoded by a single gene, say it's a biosynthetic pathway or a suite of different metabolites, then that microbe can continue on in our biologicals pipeline. So we also have a second method for discovering trait products from the soil microbes, and that's our genomics pipeline. And that pipeline is fed by a few different things here. So one thing, as I mentioned, we are sequencing all of our isolates. We're collecting pretty deep sequence on the whole genome. And so from that genome, we're using homology to then identify new members of known insecticidal and nematicidal toxin families. And we also are doing some bypassing of the isolation and preparing metagenomic samples, and we have a gene selection method that, again, uses homology to discover new candidate traits. So by having these two different pipelines, they complement each other well, and they each have different advantages that we're capitalizing on. So for the genomics pipeline, the advantage is that you're already in the ballpark. You can rapidly screen novel homologs of known insecticidal genes. And then for the empirical pipeline, where we're screening our diverse microbe collection, that has the chance to discover completely new modes of action, and it also can feed a trait or a biological pipeline. And so for the empirical pipeline to work well, it's important that we have a diverse collection of microbes. And that was one of the main reasons early on that drove us to sequence our microbe collection so deeply, is to actually understand the diversity that we were finding. So one of the things we do with these whole genome data that we get for each microbe that we isolate is we take the genome data and we identify all of the protein encoding capability within that genome. So a given bacterial genome will encode for somewhere between 3,000 and 10,000 different proteins on average. And then we can compare on a microbe by microbe basis the entire protein coding capability for each microbe versus every other microbe in our collection. And we look at that data quantitatively, and this is one way to represent the diversity in our collection, is a heat map, where we've compared each pixel is a whole genome by whole genome comparison of an individual microbe to the other microbes in the collection. And so with this, we can see, and here on this heat map, the darker pixels are genomes that are more similar, and that's why you have the really dark, when you compare a genome to itself, you get a black pixel. So this is a really high resolution look at the diversity of gene encoding capability in our protein encoding capability in our collection. And we've compared this to some of the more traditional methods for evaluating microbe diversity. And so if we took our collection and looked at just the species present in that collection, we'd capture about 8% of the diversity. And if you went a little deeper and looked at the specific 16S encoding gene there, we could capture about 31% of the diversity relative to what we capture when we look at the whole genome, the whole protein coding capability of each genome. And so a big reason why we want to understand this diversity is so that we can assay smarter. So we can prioritize microbes that we screen in our assays based on optimizing the gene diversity that we're putting through the screens. And then once we have an initial hit, we can go back into our database, find other microbes that are similar, and screen those in the assay to see if we can improve on that initial hit. That's also helpful because we can find the nearest neighbors that are inactive and then compare those genomes and see what the active microbe has that the inactive microbe doesn't have, and that's your automatic shortlist for candidates that could be responsible for the activity. So the other thing I mentioned that we're doing is another big reason why we decided to sequence so many genomes is that we are looking for new homologs of insecticidal and nematicidal toxin family genes. And of course, we're feeding all of this back into our isolation data. And so based on the presence of gene diversity and enrichment of these target genes and enrichment for hits in bioassays, that can go back and give us important information for how we should be sampling. We've identified some particular sample types that seem to be really enriched for these target genes, so we can go and try to find more similar samples. And it also tells us how deeply we should isolate from a particular microbe collection before we start sort of saturating the diversity that we find in that type of collection. So the last thing I want to talk about is just a little bit more about some of the new genes that we're finding, the new insecticidal and nematicidal candidate genes that we're finding. And one thing, I think I mentioned this earlier, is that it's for this to be a fruitful endeavor, for this genome sequencing to lead to the genes that we're looking to, we had to really enrich our collection for the species that are likely to have these genes. And so that's one thing we set out to do early on, is to enrich our collection for things like that. And one of the things that we've made the most progress with is enriching our collection for Bacillus thuringiensis genes, or organisms and genes. And so BTs are a known source for some of the insecticidal genes that are in trait products already. And in particular, these 3-domain endotoxins that kill certain insect pests are commonly found in BT strains. And so, but they have a low natural abundance. Before we started doing targeted enrichment, only about 5% of our whole collection was from the Bacillus family, and then only a really small fraction of those actually had the genes that we were interested in finding. So we have 2 proprietary methods to enrich our collection for BTs and then also for the candidate genes that we're interested in. And our patents haven't issued on those yet, so I can't go into great detail on the methods. But I can show some of what it's yielded so far. So we have about 4,000 novel BT isolates, and then we also have several hundred new candidate genes. And these are good quality genes. They're complete. They contain all the domains that they should have. And one way that we've been looking at the diversity of these new genes that we've discovered is in this phylogenetic tree, where we've taken the cry genes that had been discovered previous to AgBiome and put those into a phylogenetic tree, and then we laced in the ones that we've discovered with our 2 new methods. And we've also marked on the perimeter the activity classes. And sort of the take-home message that I think you can see pretty clearly visually here is that the sequences that we are discovering are filling in some sequence space in the different activity classes against nematodes and different lepidopteran and coleopteran pests. And then we're also filling in a lot of just totally novel families of these crygene toxin genes. And we have similar distributions of genes that we've discovered in other toxin families, insecticidal toxin families. And so we're pretty excited about this, and we're busy testing these in different systems against the different pests that they might kill. So that's all I have. Thank you for the invitation and for listening.